I'd like to thank everyone for joining today's symposium. Today, I'm going to talk about LRRK2 mutations in Parkinson's disease, which I've been working on the last 2 years. So this is the outline of my talk for today. I'm going to take you guys through about the hallmarks of the disease, some current update on Parkinson's disease, the sample set I've been working with, some key findings, and some take-home message. So Parkinson's disease is a neurodegenerative progressive disorder that primarily affects the movement. It is characterized by the loss of dopamine producing in neurons in specific region of the brain called substantia nigra, which is located in the middle of the brain. The reduction of dopamine levels leads to abnormal activity. By the time the motor and non-motor symptoms is noticeable, 60 to 80% of the neurons have already been lost. In addition to the loss of the dopamine producing neurons, Parkinson's disease is also associated with the presence of Lewy body in the brains. Lewy bodies are abnormal clumps of alpha-synuclein protein that accumulate inside neurons. The exact role of Lewy body in Parkinson's disease is not fully understood, but their presence is a hallmark of the disease, and they are thought to contribute the development of neurons. As Parkinson's disease progresses, it can affect other areas of the brain beyond the substantia nigra. This can lead to a broader range of symptoms, including cognitive decline and autonomic dysfunction. Parkinson is the second most common neurodegenerative disorder after Alzheimer's disease. Currently, 1 million people in the USA are living with Parkinson's disease. This number is expected to rise 1.2 million by 2030. 90,000 people in the USA are diagnosed with Parkinson's disease each year. More than 10 million people around the world are living with PD. PD is more common in men than women, with literature review showing that men are about 1.5 to 2 times more likely to develop the disease. The exact reason for these gender differences are not fully understood. However, the several key factors may contribute, such as hormonal differences, genetic factor, environmental brain structure and function, and the immune system differences. The etiology of the Parkinson's disease is multifactorial, involving combination of genetic, environmental, age, drugs, diet, lifestyles factors. Most of the genetic factor pinpoint in several monogenic mutations responsible for Parkinson's disease, though they only account for about 10% of the cases. The majority of Parkinson's disease, however, are idiopathic in nature. These slides represent the milestones of 200 years of Parkinson's disease research since 1800 to present date. Parkinson's disease progresses differently in different individuals. The disease heterogeneity makes it complicated, the development of one-size-fits-all cure treatment. Despite the extensive research has been done over the years, there is currently no cure for Parkinson's disease. Treatment options are available, such as medication like levodopa. It helps to manage the symptoms, but do not stop the progression of the disease. I'm not going to spend too much time on this why metabolomics matters, because Marisa Jones already explained this in her presentation earlier. So in a nutshell, in general, fields like genomics, transcriptomics, and proteomics provide an insight into what might have happened or what may occur in the biological system. However, metabolomics offer a real-time dynamic view of the biological system, reflecting the current state of the cellular process and providing a continuous and up-to-date snapshot of the biological activities. Biomarker discovery can occur in various fields, such as including genomics, epigenomics, and proteomics. Biomarker discovery helps to bridge the gap between the genotype and phenotype. NCBI PubMed search engine was used to plot the chart. Within the last 24 years, the number of biomarker search in relation to Parkinson's disease has significantly increased. It is worth noting that the time it takes to discover biomarker panel can take several years to decades, which involves multiple stages. It also underscores the importance of sustained research efforts and funding. The LRRK2 Consortium was created to assemble and study groups of people with and without Parkinson's disease who carry the mutations in the LRRK2 gene. It comprises 3 close studies, such as LRRK2 cross-sectional, LRRK2 longitudinal, 23andMe blood collection study. We received LRRK2 longitudinal study from Michael J. Fox Foundation. It contained age-matched LRRK2 mutation control with LRRK2 mutation PD, age-matched idiopathic healthy controls with idiopathic PD for 2 different time points, such as visit 2 and visit 3. So the question might arrive now, why LRRK2? There are so many other proteins also involved for in order to confirm the status of the Parkinson's disease. Well, LRRK2 encodes a protein that involves a variety of cellular processes, including signaling, protein synthesis, autophagy, cytoskeletal dynamics, vesicle trafficking, and mitochondrial function. Although alpha-synuclein was the first identified gene to be associated with Parkinson's disease, its mutation can only confirm the familial form of the disease, wherein LRRK2 stands out to both familial and idiopathic cases. So the importance of using LRRK2 genetically predisposed sample is that we can identify this individual earlier, and can we use what we find in those genetically predisposed individual in idiopathic cases. Hopefully, LRRK2 findings will help us to recapitulate it in idiopathic cases. These samples provide a window into genetic and molecular basis of the disease, allowing more accurate and effective therapies and the possible development of personalized medicine. So the sample set I have, the early onset age is 34 to 45 plus, middle onset age is 50 to 60, and late onset age is 65 to 80 plus. The biocrates Quant 500 assay was utilized with 6,500 unit, 6,500 plus unit, which is a triple quadruple MS instrument for quantitative analysis. This targeted panel offer about 500 to 600 metabolites coverage. To cross-validate, it's always a good idea to cross-validate the finding with another analytical platform, and this case is the NMR. To cross-validate the targeted findings, we also confirmed 17 common metabolites using the NMR platform. And we also did global untargeted metabolomics analysis using ThermoExploris 120 to achieve a broader range of understanding. It helps, in general, in the untargeted platform, it helps to identify 5,000 plus to unlimited number of metabolites. That leads me to my results slides. This is a heat map. This heat map analysis indicates that both types of Parkinson's disease individual with LRRK2 mutations or LRRK2 mutation and those with idiopathic controls in Parkinson exhibit significant biochemical diversity between genders. This gender-based biochemical diversity could have an implications for personalized treatment approaches and the understanding of underlying mechanism of Parkinson's disease. So sphingolipid metabolism are key components of endolysosomal pathway, which is dysfunctional in Parkinson's disease. Postmortem Parkinson's disease brain tissue analysis has shown that sphingolipid metabolism is disrupted with higher ceramide levels. This disturbance affects the protein clearing system. In our data set, compared to idiopathic cases, the LRRK2 mutations in females tend to be significantly upregulated at visit 2 compared to visit 3 than compared to in males. Glycine is an amino acid that acts as a neurotransmitter in the central nervous system. In the context of Parkinson's disease, PD, in the context of Parkinson's disease, glycine-related drug candidates are being explored for their potential neuroprotective and neuromodulator effects. Glycine concentration is also significantly downregulated in males compared to females in the idiopathic group during visit 2. In visit 3, it might have improved slightly, but it's not as upregulated as we have hoped. These groups are in treatments with levodopa. These whole cohorts are in treatments with levodopa, so which is further complicate the disease management. Phosphatidylcholine is a precursor to acetylcholine, which is a neurotransmitter that is critically involved in memory and cognitive function. In PD, where cognitive decline is a concern, disruption in PC metabolism may also affect acetylcholine levels, contributing to both motor and non-motor symptoms. Males seems to be significantly affected than females in LRRK2 mutation-associated PD during visit 3. In PD, neurodegeneration could be linked to these changes, making PC a potential marker for the disease progression. Homocysteine is a sulfur-containing amino acid that arises during methionine metabolism. Elevation of homocysteine synthesis are commonly found in vitamin D, vitamin deficiency, and polymorphism. Elevated homocysteine levels have been associated with cognitive impairment. Levodopa treatment of Parkinson disease tend to further elevate homocysteine levels due to levodopa via the catechol or methyltransferase pathway. So homocysteine levels are upregulated all conditions at both time points. However, male seems to be more affected than female in LRRK2 mutations associated PD during visit 3. In general, the area under the curve is often used as a summary metric. Area under the curve values ranges from 0 to 1, with higher values indicating better model performance. The ROC curve is above 95% for DOPA treatment, indicating that this model can be used to clinically differentiate treatments with higher accuracy. But you have to make a note that it is easy for us to distinguish between the levels of the dopamine because all of these patients are with levodopa treatment. And removing the dopamine and its related metabolites, this is the results we get with the targeted data. Not so good. Although we have identified various biochemical pathways exposed between idiopathic and LRRK2 mutated sample sets in visit 2 and visit 3, we cannot further utilize these targeted results for biomarker validation due to the significant influence of levodopa treatment. On the other hand, the untargeted data is relatively new to us. Luckily, our untargeted data, we have excluded all PD-related treatment drugs as advised by Dr. Peter Lewitt, a noted and expert and specialist in the field. By removing all the drugs, we are getting much better model. The ROC curve is between 95% and 100% for positive mode and visit 2 and visit 3. These results are irrespective of the genetic predisposition. So the accuracy of the top 2 to top 546 significant candidate biomarker ranges from 94 to 98% for both in positive mode between visit 2 and visit 3. However, we'll be very happy to with the accuracy of 94% rate, and would prefer to use a model with 7 candidates biomarker instead of 17, simply due to the fact associated cost and time. It is worth noting that in our recent delirium federally funded study, it took us 2 years and almost $200,000 to develop a method to accurately measure 19 compounds. So you do the math. If I have to choose 546 candidate biomarker, the experiment will be very expensive to carry on. I would like to focus on ionization efficiency. The reason because we wouldn't get similar results in the positive mode. Different compounds ionizes differently in positive and negative modes. By analyzing both modes, one can capture a broader range of metabolites, leading to more comprehensive understanding of the sample. Some compounds may only be detectable in one mode due to their chemical structure. Analyzing results in both modes strengthen the confidence and in the identification and the quantification of the compounds. We have also explored the negative mode results with area under the curve ranging between 87 to 91% from visit 2 and visit 3. Again, here I'm going to focus on the ionization efficiency a bit, because in the positive mode, we get about 546 hits with 98% accuracy. But for the negative mode, the number is half. The ionization efficiency in negative mode is generally lower compared to positive mode. This is partly because the negative ionization process often requires specific functional groups such as hydroxyl and carboxyl, which are less common or less reactive in many compounds. Many molecules will have natural tendency to move protons than they gain protons. Since negative mode ionization relies on the deprotonation process, which is not as favorable for a wide range of compounds, this makes it a bit more difficult to generate negative ion in sufficient quantities. The accuracy of the top 5 to top 258 significant candidate biomarker is 97% for negative modes between visit 2 and visit 3. Using this, again, I'm repeating this because it's a keynote, using a small number of compounds, we can almost achieve the same percentage of accuracy, and simply the cost and the time, as I previously mentioned as well. This brings me to my final slide for today and some key takeaway message. We are currently in the process of validating the possible biomarker candidates. It's a time-consuming, very time-consuming and laborious work, to be fair. Therefore, we won't be able to reveal the list of the compound name until this data set is published or potential future patent purposes. We have been in contact with Michael J. Fox Foundation, and they're very excited about the results, which might lead us to a possible grant. First is the method development of targeted compounds from the global untargeted data set that we have figured out. And the second is to validate those using PPMI cohorts, Parkinson Progression Marker Initiative study. Last but not the least, we are here until Parkinson is not. First of all, I would like to thank Stuart for your continuous guidance, support, and providing me with the opportunity to present the project. Thank you to my work colleagues specifically for helping me out on this project. Thank you to Team Woojin for the exceptional bioinformatics analysis. Special thanks to Peter, Nick, Stephen, Patrick, and Michael J. Fox Foundation. A huge thank you to the sponsor. Thank you all for listening, and I'm happy to take any questions you may have.